Hi guys, welcome to this Sunday YouTube session. Just give me a quick thumbs up or hello or something just to let me know that you can hear me okay. And yeah, how is everybody this evening? Anyone got exciting plans for Christmas? Or is anybody stuck at home, self-isolating? I'm in that boat. Although last day today, so I can finally leave again tomorrow, which would be nice. Yeah, anyone else in the same situation? It seems like half of London has COVID at the moment. Winter Wonderland, oh fun. Yeah, I mean, I've been meaning to go to that actually. It was cancelled last year, wasn't it? But yeah, it'd be good to go this year maybe. Sorry you've had COVID as well. Yeah, sorry to hear that, but it's good that you're finally in the clear. Yeah, what happens with, so in theory, I think I can leave the house tomorrow. It's been over 10 days tomorrow, but I did a lateral flow this morning and it was still showing positive. So I'm not sure. Apparently from what I was Googling is it might stay positive for a while, even if you're not contagious. So yeah, I'm hoping I can still go out tomorrow. Yeah, well, I had it, I think more or less like got rid of it now. It's been enough days. Cool. Do I think there'll be another lockdown? Yeah, I don't know. I really hope not, but the cases are going up. We'll have to wait and see whether the hospitalizations also rise. Obviously the cases are like massive right now, but if that doesn't transpire to spike in hospital admissions and well, the worst is obviously deaths. If they don't go up, then I don't think we will do one. But if they do go up, then yeah, who knows? Could be. Anyway, yeah, I guess just in the meantime, follow the government advice and try and stop the spread, and then touch wood, that will be enough, and we won't need to do a full lockdown. Yeah, but as well, yeah, so many people right now. Like I feel like half my friends are all isolating at the moment. So anyway, yeah, let's make a start. So I'll share my screen. So you should be looking at a PowerPoint now. Everyone see it? Masks don't work. Oh, probably shouldn't get into this now. The TLDR on masks is they are very effective at stopping you spreading it to other people. They are less effective at stopping you getting it. Yes, but certainly altruistically, they work very well. Yeah. Yeah, exactly what B. Hughes is saying. Anyway, yeah, let's not get into all that now. We're going to talk about protein synthesis today. So I can't remember what we did last month. I feel like it was something similar. Did we do DNA? It was quite a while ago now, so can't really remember. But anyway, so we're doing protein synthesis. We're going to look at basically transcription and translation and the steps involved in that. Can you go from an E grade to an A grade? Yeah, definitely. Lots of people have done it. It's not easy, but it's definitely possible. You've got plenty of time. If you start now, that's loads of time to really pull around a grade. So yeah, a bit about me. My name's Alex. I'm the head of biology here at SnapRevise. I've got a degree in biology and a teaching degree as well. Been teaching and tutoring for the last 4 or 5 years. So we're offering a little special promo deal today, Christmas promo. Oh, we should have actually given it a themed Christmas code name. Oh well. So the code is back to school 21, and this is going to give you 25% off your first month. So I'll talk you through the website a little bit more at the end of the session. So So yeah, I guess without further ado, let's dive right in. So those of you who've done one of these before, you will probably recognize this. We normally start these sessions with a little pyramid for revision. So these will be questions relating to topics that we've studied in the last few weeks, in the lead up to this session. Nice. So first question, how are sister chromatids different to homologous chromosomes? We're not going to do mutations today. No, that's another lesson. Can't do it all in one session, unfortunately. So what would you say for this question? What's important to do in the first 2 weeks of the Christmas holiday? Honestly, take some time off, take maybe a whole week, the time between Christmas and New Year, maybe. Well, if you've been totally slacking this term, then maybe don't take time off. Use the time to revise. But if you have been actually working, you need to have some time off, because the next holidays, February half term and Easter, they're not really going to be proper holidays for you. If you're year 13, you need to be revising in those ones quite hard. So I would say in this one, enjoy the time off. If you were totally slacking this whole last term, then maybe you don't need a break and you need to actually catch up, use the time to catch up. But if you have been working hard, yeah, I would recommend taking some time off. And yes, nice answers. So sister chromatids are identical. Homologous chromosomes are same genes, but different alleles. So we could say for this one, identical, i.e. same alleles. Essentially, you have 2 chromosomes for each type of chromosome. So we've got 23 pairs of chromosomes in humans. So let's say this is chromosome 1, and then chromosome 2. You see how you've got 2 of each one? We call these maternal and paternal. You get one from your mother, one from your father. That's how it works for sexual reproduction and organisms that reproduce that way. You get half of your chromosomes from your mother, half from your father. So they're called maternal and paternal quite often. So these are homologous chromosomes. The 2 copies, one being from your mother, one being from your father. They have the same type of genes at the same loci, same positions. So say there's not just one gene for hair color, but let's pretend there is. The gene for hair color from your father is on that one, and the gene from your mother is on that one. Maybe you get a brown hair gene from your mother and a blonde hair gene from your father. So they're going to have different alleles. Sister chromatids are when you have replicated your chromosomes. So you have 2 identical copies of this chromosome and 2 identical copies of that chromosome. These are the sister chromatids, and these are the homologous chromosomes. That's one of the, I feel like I've talked about this before in one of these sessions, but it's because it's super, super important. You can't really understand meiosis without having that really clear in your head, and also a lot of the genetic stuff you do in year 13. So you need to have that completely dialed down. But it seems like most of you do from what the answers you were giving me. Okay, next question. Define the term degenerate in terms of DNA, not in terms of the insult. Yeah. So more like some of you wording that a little bit strangely. Just, yeah, I would say more like something like multiple codons can code for same amino acid. Yeah, or something to that sort of idea. This is for all exam boards. We do questions from from all the main UK exam boards. So also if you do IB, it's pretty similar as well. So what tRNA-based sequence would go with the following mRNA sequence? Yeah, Ruth, exactly. Because of the code being degenerate, this can lead to silent mutations, which don't have any, like, you can't see any difference, even though there has been a mutation. Yeah, exactly. U, G, C, A, U. So I haven't lined mine up perfectly. But yeah, it's already, we're going from RNA to RNA, so the U's are already in there, and we just keep them there. Nice. What's the difference between the exam boards? Slightly different. Some of the topics are a little bit different. Most of the core material is almost exactly the same, but there are a few topics which one exam board will do that the other one won't do. There'll be some examples of that today. I'll point them out when we get to them. Then, so how is the secondary immune response different to the primary immune response? This is just like a short answer, not like a full explanation. This is usually worth 2 marks in an exam, and it's quite a common question. There's 2 things they're looking for here. This isn't to do with the specific versus non-specific. This is, both of these are specific immune responses because they both involve antibodies. So we would say, kind of run out of space. Yeah, secondary is quicker. This is because you don't do clonal selection or clonal expansion, but we're not going to get into full details of that. But if that means something to you, that's why. Secondary is quicker, secondary, and produces higher concentration of antibodies. And yeah, as some of you guys have said, this is all to do with memory cells. You have the memory cells already if you've gone through the primary response. Therefore, you can use those memory cells to start producing plasma cells, which make the antibodies much more quickly. So you can produce this big concentration of antibodies very quickly. The idea is it's quick enough so that you don't really get ill. You don't really get symptoms because you've created the antibodies before the pathogen can get a proper foothold. So yeah, you get secondary immunity, or you'll do the secondary immune response if you've already had the illness or if you had a vaccination for the illness. So that's the that's the benefit of getting a vaccination. So what has become apparent is that the COVID vaccine doesn't stop you necessarily getting ill. It might stop you getting ill, but you could still potentially catch COVID. We did hope at one point that maybe it would be like completely stop you getting the virus, but it looks like you can get it again. But what people are noticing is the symptoms are usually much milder if you've been vaccinated, because you're not producing as much virus and because it doesn't have as greater effect. So yeah, there's still benefit. Kind of looking at the time, but I'm not going to explain this one, but I'll just quickly like put how you would do it. So yeah, basically, if you've got 15 molar solution and you need to make 11 molar solution, and you want 20 centimeters of it, you do 11 divided by 15. So this is your desired strength divided by your starting strength. And that's going to give you a decimal somewhere between 1 and 0. So in this case, I think it's 0.73. Then you just times this decimal by the volume that you want. So 0.73 times 20 equals 14.6. So to make 20 centimeters cubed of this 11 molar solution, you need this much 15 mole solution, and then the rest you just top up with distilled water. So like 5.4 water. Mix it up and you'll have 11 molar solution. And you can obviously do that for any you can plug in any numbers to get that to work. So let me just catch up any questions I haven't answered. Yeah, DNA always has thymine, RNA always has uracil. Exactly. Cool. I think that's everything. So yeah, let's dive in. So we're going to look at transcription. We're going to look at alternative splicing. This isn't in all exam boards, and I can't remember exactly. It's definitely in AQA. I think Edexcel talk about it a little bit. They talk about splicing at least, and then OCR don't actually talk about it at all. And then we're going to look at translation as well. How do you get 14.6? That's this is this decimal times the starting, this times the desired volume. Oh yeah, and sorry, the 5.4 is like what you would add to make it up to 20. So yeah, as Zara was saying, 20 minus 14.6 is where you get the 5.4 from. AQA Edexcel a little bit, but not OCR. Oh, turning on the Alexa. And then yeah, hopefully we'll have time for translation. We'll see how we're getting on. These ones on YouTube take a little bit longer normally. Alexa, stop. Don't know why it's setting it off. So these are the spec points. Not going to really talk about them today. You guys should all have a copy of your spec. So let's start with transcription. So I guess like, firstly, why do we need to do transcription? We'll look at like how it actually happens. But yeah, why do we need to do it in the first place? CIE and AQA are quite similar. Yeah, but there are some differences. Like CIE do loads more detail on plants, and they like focus a lot on, you'd have to look at a lot more actually like microscope slides of the respiratory system. So there are some differences, but those two are actually relatively similar. Yeah, so we need to get the genetic code needs to be or needs to reach, should we say, reach ribosomes. These are outside of the nucleus. DNA to too large to leave. Therefore, messenger RNA transfers the genetic information. And then obviously transcription makes the mRNA. So the terminology is kind of logical, like messenger RNA is the one that takes the message from the nucleus to the ribosome. Transcription, script is like writing. So transcription is writing the messenger RNA, writing the message. And then translation is turning the message into protein. So you're translating genetic information into actual amino acid sequence. So yeah, if you ever like get the two confused in your head, just think like transcription, script, scribing, writing, translation, sort of changing one thing into another. So yeah, let's look at how this works in a little bit more detail. So the enzyme involved is RNA polymerase, not to be confused with DNA polymerase. I think some of you mentioned there. DNA polymerase is the one used in DNA replication, whereas RNA polymerase is used in protein synthesis. So RNA polymerase is the one that sort of unzips the DNA. It's not the best terminology to use. You could say like separates the two strands by breaking the hydrogen bonds in between them. If you recall DNA replication, you'll remember there's another enzyme involved in that. That's helicase. So in that case, helicase does the job of breaking the hydrogen bonds. In transcription, RNA polymerase actually does two jobs. It breaks the hydrogen bonds, and it also joins the bases together. So it forms the phosphodiester bonds. It's a multi-talented enzyme. It does both jobs at the same time. So yeah, it moves along, joining the phosphodiester bonds. Located between the nucleotides, and then we get a strand of, it will be pre-mRNA initially, and then you will do splicing to form mature mRNA. What's the difference between RNA and mRNA? mRNA is a type of RNA. Sorry, I'm going to have a few coughs because, yeah, well, like I said, I had COVID last week and it's still a bit of a, yeah, still got a bit of a cough. I know it's annoying if you've got headphones in. I'm trying not to cough directly into the microphone because I know it makes it like super loud. It's quite annoying. But yeah, I'll do my best. Yeah, so as I was saying, mRNA is a type of RNA. There's different types. There's ribosomal RNA and there's tRNA. So that's the main difference. It's a type of RNA. Specifically, mRNA is single-stranded, as you guys are saying, and it's the length of the gene, however long the gene is. The mRNA is the same length. What else to add? Yeah, so we copy the template strand. The one we don't copy is called the coding strand. Or you might have heard of the terminology sense strand. And the template strand is called the antisense strand. Now, at first glance, these names might seem a bit weird because we're copying the antisense strand rather than the sense strand, or we're copying the template strand, we're not copying the coding strand. The reason for that is because when you copy it, because of complementary base pairing, you get the opposite of the strand you copied. So say there's like a base A here, or maybe A is a bad example because it's going to change between thymine and uracil. Let's say there's a G here. There's going to be a C on your copy. So if this strand, the coding strand, aka the sense strand, has the base sequence you need, then you need to copy the opposite strand, because if you copy the one you want, you're going to get the opposite. I feel like I'm using the word opposite a lot, but hopefully that's making sense. Yeah, so you copy the one that's complementary to get the same as the sense strand. So this is where the actual gene is located. I'll put a note. Contains genes. Whereas this one is like the complementary opposite to the genes. So let's go through. If you copied the coding strand, you would get a different protein, and it almost certainly wouldn't have the same shape or function because it would be a totally different protein. So yeah, you definitely couldn't copy the same one. Yeah, it's kind of like a mirror effect, like how a mirror reverses the image. Exactly. So if you mirrored the image again, it would look the same, like flipping it once, flipping it twice. Exactly. So what can we say? Let's add some like brief notes. So RNA polymerase breaks H bonds between strands. Binds. Oh no, let's say we'll do that later. So free, keep changing my mind. Free floating RNA nucleotides bind to the exposed bases via complementary base pairing. I'm just going to put CBP because there's not enough room to write that out. That's not a proper acronym though, so don't write that. So joins via complementary base pairing, then RNA polymerase moves along, joining nucleotides with phosphodiester bonds. So the RNA polymerase is joining the nucleotides together, moves, probably should put this earlier, down the template / sense strand, no, antisense strand. Then pre-mRNA strand detaches and is ready for splicing. Oops. to form mature mRNA. You don't usually have to write out mature mRNA. You can just say like RNA and people assume it's mature. Yeah, I see some people are asking about pre-mRNA. So if you're OCR, you don't really talk about this at all. So that's maybe why you haven't heard of it. Amrit, yes, exactly. RNA polymerase does both. Helicase is only in DNA replication. This is a really common mistake. I've actually seen it in textbooks sometimes as well. So yeah, I don't think you'd be penalized in a mark scheme. You just probably wouldn't get a mark for it. Why in the picture is RNA polymerase attaching the? So it's not it's not attaching it. Yeah, maybe the diagram is a bit misleading. So essentially these bases, they match up by themselves just due to complementary base pairing. And then what RNA polymerase does is joins these phosphodiester bonds. This strand, these are just hydrogen bonds, so they can be broken. So the actual mRNA strand will just like be removed at the end. Yeah, there's no such thing as RNA helicase, exactly. Yeah, and DNA replication, there's no RNA involved. Anytime you see RNA, think protein synthesis or transcription and translation. So yeah, the difference between pre-mRNA and mature mRNA is that the introns are still in pre-mRNA and they're not in mature mRNA. Exactly. The whole strand is not unzipped, only the section that you're needing to copy. So, so say like your chromosome is however many, like maybe 30,000 base pairs in length. If you need one gene that's only maybe like a few thousand base pairs, you just unzip that little section and copy it like that. You don't need to you don't do the whole thing. Whereas obviously in DNA replication, you need to move the whole way down to get a whole copy. Yeah, exactly. So DNA replication, creating an identical copy of the DNA. The point of that is to do cell division, because if you want the cell to divide, if you want one cell to become two cells, each of those cells needs to end up with a copy of the DNA. So you need to replicate your DNA so that both the two new cells have a full set of DNA. Cool. So let's try some questions. DNA replication is for both mitosis and meiosis. Yeah. Anytime you need to replicate a cell, you need to divide the DNA. So it's also, DNA replication is also true for like binary fission in prokaryotes. So yeah, both. So how are we feeling with that? The main thing you would say, RTG, for what the RNA polymerase does is it joins the phosphodiester bonds between the RNA nucleotides, which have paired up via complementary base pairing on the template strand. Cool. So yeah, we're going to do some past paper questions just to see how you guys are getting on with this stuff. Annoyingly, the first one has the answer on the screen. So it'll be nice and easy for you guys. And yeah, good to see. Mostly ones and smiley faces there. Nice. So this shows a short sequence of DNA bases. What's the maximum number of amino acids which this DNA could code for? So yeah, it is 9. The way you do this is just count how many there are and divide it by 3, because DNA or RNA is read in 3s. So you can just go boom, 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 like that, and then count up 1, 2, 3, 4, 5, 6, 7, 8, 9. And that's our answer. However, so that's the maximum. However, it's likely we would actually get less than 9 amino acids if that was our gene. So what's the reason for this? There's a few things you could say here. So it could contain start or stop codons. Yes. There's also something else you could say. A few of these saying the same thing. You can't say degenerate here, because degenerate would still mean there's the same number of amino acids. So that wouldn't be an accepted answer. Again, yeah, we're going to assume there's no mutations, so that wouldn't count. But Huda, yeah, you've hit the nail on the head there. There might be introns in there. So it could be introns in the DNA do not get turned into amino acids, so they're not expressed. So yeah, hopefully that makes sense, and hopefully that also makes sense why you can't say degenerate, because that just means the amino acid is different. Or like a mutation would also like change the base sequence. Well, I guess no, if it was a deletion, it would be less, but yeah, we're assuming there's no mutations. If you haven't heard of introns and exons, then it'll be explained on the next page. But if you're not OCR, sorry, if you are OCR, you don't actually need to know about them, but I can explain it quickly anyway. Okay, so next question. A piece of DNA contained 74 base pairs. 2 strands of the DNA were analyzed to find the number of bases of each type, and the results were shown in the table. So this is a super common style of question you get when you look at DNA. So these questions, they'll either tell you how many base pairs in total, or they'll tell you the number of base pairs. Sorry, they'll tell you how many, or they'll tell you the percentage. And obviously percentage is out of 100. If they tell you how many, you need to work with that number. Start and stop codons. Yeah, if you have like a gene like this, the first one actually doesn't code for an amino acid. It just tells the tRNA. It tells the ribosome, that's the like starting point. And then the last one also doesn't code for an amino acid. It tells the ribosome that you've finished the protein, basically. You finished copying it. So start and stop codons don't correspond to amino acids. So yeah. So what can we say for this? So number of bases, we'll fill in what we can just by copying off the complementary pairs, and then we have to do a relatively simple maths calculation to get the last numbers. So 2 strands. If I've got 26 C bases on strand A, then I'm going to have 26 Gs on strand B, and then vice versa. 19 here means we have 19 here, and 9 As here mean that we have 9 As over there. Nice. So if that's the case, we now need to work out how many A's are on strand A and how many T's are on strand B. Well, they're both going to be the same number. So all we need to do, and you guys can probably work this out, is we do the total number, 74 base pairs, minus 9. Well, actually, I should probably write it like this. Minus 9 plus 19 plus 26. Which gives us what? 20. So now if you added this whole row up, like this plus this plus this will equal 74. That's our base pairs. If it was percentages, you would just make sure that they add up to 100 at the end. That's the only difference. And yeah, like I said, they can do either way. They're both like equally common, to be honest. Okay, so next question. What is the role of RNA polymerase during transcription for 1 mark? So someone asked about this a few minutes ago. Where's the 74 from? Yeah, exactly. In the question. If they don't tell you how many bases, it's probably because it's percentages instead. Yeah, so you can say it breaks the hydrogen bonds. However, I would probably just say that it forms phosphodester bonds between RNA nucleotides. That would be fine. Just forms phosphodester bond between RNA nucleotides. Or actually, yeah, catalyzes is an even better word. Catalyzes the formation of the phosphodester bonds. You could add on template strand if you wanted. Obviously not like the ones in the template strand, but on the RNA nucleotides that are on the template strand. DNA helicase does the hydrogen bonds, but only in DNA replication. That's why it's not here. You can't say joins two nuclei, no. Nuclei is like the plural for nucleuses. So yeah, that would be incorrect. Doesn't form hydrogen bonds. Hydrogen bonds form spontaneously between the complementary base pairs. So it breaks hydrogen bonds, and hydrogen bonds form by themselves. They don't need an enzyme. Yeah, Marija, no, that's only in DNA replication. Yeah. Could you say joins nucleotides? I would say the name of the bond usually is necessary to get the mark. So forms of phosphodester bonds between nucleotides would be the best answer. Cool. So Next question for 2 marks now. What is a codon? Yeah, I would agree. That is a good answer from Hajar there. In fact, yeah, not much to add for that. So, on mRNA strand, that's first mark. The code for an amino acid, or I guess you could say stop. Trying to work out what's like something's rattling. What it is. Okay, I stopped it. The codon is not the same as a triplet. So actually, this question was a quite generous. If you said DNA here, they allowed it, but codons are technically only on messenger RNA. So, like, this is our DNA strand. These things on DNA, they're called triplets. The same thing when you copy it to mRNA. So say like this would be UAU on mRNA. These would be called codons. And then the anticodon, which would be AUA. Oh, sorry. So these anticodons will be found on tRNA. Kind of got these like flipped around a little bit, but yeah, triplets are on DNA, codons are on mRNA, and anticodons are on tRNA. So that's the difference between those three. In terms of the sequence, you'll find that triplets and codons are complementary, and also codons and tRNA are complementary. So actually, triplets and anticodons are the same, although the A's have been swapped with U's because one is DNA, one is RNA, but the actual DNA sequence is the same. Yeah, I think that's everything I want to talk about on this page. So let's talk about splicing and alternative splicing as well. So when you initially copy the gene, you're going to copy these sections called introns, which actually don't code for the protein. So you need to get rid of those before you translate the protein. So let's say we copy this. So now we're going to make a pre-mRNA strand. That's still going to have the introns and exons are going to both be in there. So we need to cut out the introns. So splicing removes the introns. It is carried out by the spliceosome. I think that's all we need to talk about that for now. So removes the introns on pre-mRNA and is carried out by the spliceosome. Yeah, if your OCR you can kind of zone out a little bit, but I guess it's interesting, and it's not that much extra to learn. Only eukaryotes do this. Yes, prokaryotes don't have introns. Yeah. So that's splicing, and then we have something called alternative splicing. So this allows multiple different proteins to be made from the same gene. Oh, that's kind of like both points in one. So say in this gene, we have 4 exons. We've got exon 1, exon 2, exon 3, exon 4. We don't just have to stick them all together in one order. We could arrange them like this, 1, 2, and 3. That gives one protein. We could do 1, 2, and 4. That would give a different protein. We could do just like a 1 and a 4. This would give us a shorter protein, protein C. We could do all 4 of them. 1, 2, 3, 4. D. So you see there's a whole bunch of different proteins we could make here just by arranging the exons in different orders. So that idea of arranging them in different orders, that is called alternative splicing. And yeah, for people who are asking about what's the difference? The exons code for amino acids. So the exons we can say are expressed. The introns are removed. So the introns are non-coding parts of the DNA. AQA do alternative splicing. Yeah. So you do need to know this. There's not much to say about it really, just that, yeah, you arrange the exons into different positions, and that can allow more proteins to be made from one gene. And this can be actually quite significant. So there's a gene in fruit flies, one gene that actually can code for over 38,000 different types of protein, all because of alternative splicing. So that gene is massive, and there's like hundreds of different exons in there. So you can rearrange them into a very large different number of proteins. So yeah, so far we've found around, well, just over 20,000, I'll just say 20,000 for now, 20,000 genes in the human genome. But because of alternative splicing, we can make around 100,000 different mRNA transcripts. Oops. So, and we can actually make even more protein. We can make even more different types of protein because we can actually modify the proteins once they've been synthesized. We can like add carbohydrates to them to make a glycoprotein. We can add lipids to them, make a lipoprotein. We can chop amino acids off to make them a little bit shorter. We can still, yeah, we can still modify them further. No, the sense strands, exons and introns are not to do with the sense and the antisense strand. Only the sense strand contains like the actual introns and exons, but they're both on that strand. The antisense strand is essentially gibberish. It's like a mirror image of the coding strand, which has both introns and exons in there. Yeah, so the advantage of this, make a lot of different types of protein. Yeah, that's the idea. Yeah, we make all these proteins naturally. Yeah. So the actual total number of proteins is closer to 1 million different proteins. We don't know for sure though. This is like we haven't documented every protein that the body makes. We're finding new ones all the time. So the vast majority of these are enzymes. And yeah, we discover more every year. So how are we doing for time? Let's quickly do these questions and then we'll end there. Yeah, introns they're bases, but they don't code for protein. They have roles in like helping to regulate gene expression and with alternative splicing, but they don't they don't actually directly make protein. Yeah. So describe and explain what happens in step 2 on figure 1 for 3 marks. And although it's 3 marks, you can actually get the marks with relatively few words. So step 2. So step 1, hopefully you've noticed, is transcription. This is a DNA strand, and this is a pre-mRNA strand. And then this one's a bit shorter, you'll notice. So this is mature mRNA. And step 2 is splicing, removal of the introns. So splicing has occurred. Introns are removed. This forms functional proteins. If you don't remove the introns, the proteins won't work properly. They will be the wrong shape. So yeah, just checking some of your guys' answers. Yeah, so a few mistakes in there. So it's not translation, that's later. So translation would be step 3. Because if you notice, for steps 1 and 2, we're still inside the nucleus. Translation occurs outside of the nucleus, down here. So this would be the growing polypeptide chain. And this, I think this is supposed to be a ribosome here. This is obviously a tRNA molecule. And then, oh wait, we're suddenly answering, maybe you're answering this question. So explain what happens in step 1. This is transcription, where we go DNA into pre-mRNA. Using not DNA, RNA polymerase. Something like that would be, yeah, worth 3 marks. So yeah, 1, 2, 3, 1, 2, 3. So like I said, don't actually need to write that many words to get full marks here. You don't have to do it in the past tense. No, not necessarily. Splicing in eukaryotes because only eukaryotes have introns. Correct. Yeah. Nice. Any other questions? Just checking. Yeah. So yeah, and splicing, you could add maybe here at the spliceosome. But that's optional. Don't need it for the mark. Cool. Why don't prokaryotes have introns? Yeah, good question. I'm actually not exactly sure why they don't. It's obviously like the way that they've evolved, but yeah, if you don't have introns, you can't do alternative splicing. So this limits how many different proteins you can make. So it's a bit of a disadvantage. Prokaryotes can probably get away with it because they're relatively simple organisms. They don't have that many genes and they don't need to make as many different proteins. If you're a multicellular eukaryote, you need to make a lot more different proteins you're a lot more complex. So you need to be able to make all these different enzymes to help with your metabolism to survive. So that would be my sort of like guess, but I'm not exactly sure. But yeah, feel free to look it up and share your results with us next time. So yeah, we didn't actually have time. We don't have time today to get onto translation. I didn't think we would because it's quite a lot to get through in one session. So yeah, feel free to, if you wanted to carry on with this, we've got some videos on this you could look through. But yeah, that's basically yeah, it for today. Before you go, I just wanted to talk about a few different things. So firstly, we've got more of these web classes. So set a reminder, subscribe to our channel. The next biology one, I think, yeah, on the 9th of January. So it'll be in 2022. Exciting. Then, yeah, a bit about our courses. So if you enjoyed the session today and you found it helpful, then we actually run loads more of these sessions. We just don't put them on YouTube. So we do the YouTube ones once a month, but we do these classes 5 times a week actually on our website. So if you want to come along to all those sessions, then you can sign up to a course. Also in the course, you don't just get that. So you get the lessons, but you also get access to the videos, and this will track which videos you've watched. You'll get smart quizzes. These are really good. You watch a video, you do a quiz relating to the video, and then if you get something wrong, it will link you back to the correct part of the video that is telling you the information. So it's like super easy to fix any gaps in your knowledge and to like diagnose where you're getting stuff wrong and to clear that up. There's also some condensed revision guides. These are basically like cheat sheets for cramming all the information in one little one small, sort of like small area. Can't talk. It's been an hour, and my brain's done. This is my last lesson before Christmas, so I feel like I've slightly mentally checked out. No. Anyway, so we've also got these exam walkthrough videos. Again, these are really helpful, especially as you come into sort of main revision mode. Knowing the content is the first step, but you also need to be able to like score marks. You need to have good exam technique, and part of that is like looking at the command words, and a lot of that is just doing practice and yeah, sort of looking at mark schemes, seeing how they award the marks, and you can kind of memorize some of the key bits. So those will really help with your exam technique. And then just some extra questions. So these are additional questions that are similar to the actual past paper questions, because what you'll find quite quickly is you'll have done all your past paper questions, and there'll be nothing that you haven't seen. So you can't really test yourself on a question that you've seen 2 or 3 times, because even if you don't really remember it, some of it will stay in your memory. So it's not really like the same when you keep doing the same questions. So we've made different questions that you can attempt and graded them easy, medium, and hard. So super easy to revise. So I was going to show you the website. I feel like I don't really have to. You can kind of like just check it out yourself. So the website is, maybe I'll quickly show you. So just as a reminder, there's a 25% off discount code for this month. And let me open up the website. So this is the website. These are the courses I was talking about. Then, come on, how you do this? Oh, is it also? I can't remember if you can use the free trial and the discount code. Probably. Anyway, check out your subject. So, like, let's do A-level biology. Oh, wait. Okay, I won't actually sign into my account because I don't have a keyboard right now. So yeah, anyway, that's the website, snaprevise.co.uk. They're the courses. Check it out, use the discount code. I think you can also get the first month free, but not 100% on that. And yeah, I think that's more or less it. So I'll stick around for a few minutes just in case there's any questions. But other than that, thanks for joining. Have a great Christmas and New Year's or anything else that you're celebrating. And yeah, hopefully see you on our website, on our platform. And if not, see you in the next YouTube session on the 9th of January. Hajar, yes, they are a paid package, but yeah, I would say for what you get, I think it's around like £40 a month. Not 100, again, not 100%, but you get like up to 20 hours of lessons a week for that. No, sorry, 20 hours a month. Well, more like 25 hours a month. So you compare that to tutoring, which is like £50 an hour minimum, really, for a good tutor in London. Yeah, it's a total bargain. You're getting way more for your money, and plus all the extra stuff. So yeah, it's not free, but I think it's the deal of the century. Yeah, revised splicing, don't have time to go into in any more details than we just did, to be honest. Again, there's on our YouTube channel, there's some more videos on splicing. So check out the actual, not the web classes, but like on the YouTube channel, just type in SnapRevise splicing, and there'll be like a detailed video that will go through it there. Yeah. Any other questions, or you guys all good? Cool. Yeah, I think we'll end there then. So yeah, Merry Christmas. And yeah, stay safe, try and avoid the COVID. And yeah, see you in 2022. Cool. All right, bye guys. Bye.